Hello, I'm Tony DeMaria from UC San Diego, and I'm pleased to be the moderator for this session on cardiac stem cells. And there's been a great deal of excitement about stem cells for cardiovascular disease, and people ask me why. And on the left, you see the major anatomical causes of heart disease, and on the right, potential therapies. And you'll see that for most diseases, we have pretty decent therapies. We have stents for coronary disease and very good prosthetic valves and good surgeons and defibrillators for arrhythmias. But if the muscle, the heart muscle itself, is destroyed, then we don't have very good therapy. And this slide shows an example of that. This is the kind of patient we were worried about. On the left is the heart of a normal individual, and on the right, a patient with advanced heart failure where the muscle is destroyed, the ventricle is dilated, and it's poorly contractile. Of course, to apply stem cells, there's a number of questions we need to ask, and for many of them, we still don't have full answers. Why do these stem cells remain undifferentiated and self-duplicating? What triggers them to become the organ that they ultimately become? Nevertheless, we proceeded on some clinical trials. This is just one of a whole host. I show you quickly because I happen to be involved, where the use of a stem cell product actually enhanced an endpoint of death and cardiovascular hospitalizations in heart failure patients compared to controls. But of course, then the question is, how did this come about? And we would love to think that there's tissue regeneration, but unfortunately haven't been able to find evidence of that. So increasingly, we've looked for other mechanisms, paracrine mechanisms, where growth factors might be affected, or proteins that would enhance blood vessel development, something that would modulate immunity, and look for alternate mechanisms and alternate applications of stem cells. And today we have some spectacular discussions of the use of stem cells in cardiovascular disease and medicine that is clearly enhancing our understanding of the application of these cells and providing new applications for them. So the first presentation will be by Deepak Srivastava on cellular reprogramming approaches for heart disease, followed by Mark Mercola on induced pluripotent stem cell derived cardiomyocytes for predicting and removing drug cardiotoxicity. And there's lots of drug cardiotoxicity that we're encountering. And lastly, Deborah Lieu on the quest for pacemaking cardiomyocytes to engineer bio-pacemakers.
The next session is going to be about cardiac stem cells, and we're delighted to be able to welcome Pat Farrant, who has a really interesting story and is going to be talking to us about heart disease and his involvement in stem cell research. Pat, your introduction to the world of stem cell research and to the world of heart disease came rather suddenly, didn't it?
Yes, it did. Not exactly a planned event. I had been ignoring some warning signs and probably was not paying attention to what I've learned is a vital component, which is genetic base to heart disease. My father had passed away at age 61, and that's what I experienced. So I learned how to get fast action in an emergency room. You just go in and say, I'm experiencing chest pains. I'm 61 years old. That's the same age my father died. So right away, I got into the hospital, and shortly thereafter, I guess I crashed and their blood pressure dropped, and they had to do surgery at midnight till 5 in the morning. I told at one point I was down to my last 2 hours. as a main artery, the LAD, left anterior descending artery, had closed, and but came through 5 hours later, and that was 15 years ago.
Wow. Well, we're delighted you're here to join us. I know since then that you've been really active and engaged in kind of advocacy and support groups for heart disease. Talk a bit about that.
Yes, indeed. Working closely with my cardiologist, she felt I was making a spectacular recovery, and she said I really should talk with other heart patients and heart disease in general, but other heart patients about what to expect in recovery and what they have to do to get their lives back. So I called the American Heart Association locally here in San Jose, and they asked me if I like to lick envelopes or if I like to get involved in fundraising, and neither one of those particularly appealed to me. So they said they had an affiliate organization called Mended Hearts, a heart patient organization. We currently have about 40,000 members. So I became involved. We visit heart patients, 250,000 heart patients throughout America each year. I became involved at the local level as a chapter vice president, president, and then regionally as a regional director of the area, and then and then I served on the board of directors as a vice president and was elected to be executive vice president of about a 40,000 member organization. We primarily visit patients in their rooms. We just introduce ourselves as saying we have something in common with you. We've had a personal experience with cardiovascular disease.
And I know that one of those experiences where you were in the room, you saw exactly how difficult it is for patients to make decisions. I mean, there's a real time pressure, obviously, because of the heart condition, but also conflicting information. So tell us that story.
Well, it was in an innocent way that I happened to be in this patient's room when the surgeon, the cardiovascular surgeon showed up, cardiothoracic surgeon, excuse me, and he was the patient. They hadn't decided on what the course of action was going to be or the treatment. So he gave quite a presentation on the benefits and values of open heart surgery. And I happened to be back in that same patient's room where later in the day, an interventional cardiologist came in and went through all of the benefits of an intervention by putting a stent. And really, I felt wasn't giving, in terms of informed consent, of giving the patient the data, the information they needed to make a decision. It sounded almost to me like they were pitching both of the things that they did, one over the other. And the patient actually asked me what I would do. Well, obviously I can't give medical advice. And I told him he needs to talk with a cardiologist because he was, it had to be decided that the treatment was going to proceed the next day. And I didn't have a chance to talk with a cardiologist and was put in a very awkward situation, I felt, and also was somewhat in a bit of a drug haze with the medical treatment he was giving. So it made for, I felt just a kind of an awkward situation of two different procedures being promoted to them and then saying, so now what do you want to do?
Yeah, obviously it's a critical time, obviously, for the patient, and obviously informed consent and good information is a really challenging thing for someone to be able to grasp in that situation. Since then, you've become involved with a CAP, what we call a clinical advisory panel, helping guide from the patient's perspective some of the heart disease research that we're funding. Talk a bit about your experience with that. I mean, how important do you think the patient's perspective is?
I'm on the board there, and it's a bit of a difficult situation. And these are all cardiac researchers, highly trained, highly educated. And in fact, I started out by thanking them for studying hard and getting a good grade in organic chemistry, which probably helped them get into medical school. And I thank them for their choice for the field of study. Medicine and furthermore cardiology. So I said, speaking on behalf of some 40,000 patients, we thank you for how you've committed your life doing research in cardiovascular disease and heart failure. I have a difficult time following a lot of what they're saying because it's all better, and they understand that I'm, just an average person without medical training, but I try to look up as many words as I can as they're talking and follow what they're saying. Heart failure and what they're looking at in terms of regenerating stem cells, regenerating cells for stage 2, stage 3 heart failure, damaged hearts, isn't more of a heart disease person, not a heart failure person, but I'm following what it is that they're, how to design the research and all that's involved, and also have learned to appreciate the various steps that have to be taken to get FDA approval for human trials. This particular study out of Stanford isn't at human trials yet, but we're certainly I wouldn't be a candidate for that because heart failure is not my thing. But, you know, I guess one thing that came up is that when they were prior to getting to human trials, I said, well, I just asked them out of curiosity, I said, what if there are some complications here in terms of the patient having complications following these stem cells and involved hospitalizations, et cetera. So I just asked them, who would pay? And I could kind of tell by the nervous looks going around that room, that hadn't exactly been established yet. I mean, it's funded by a grant from the state of California and the PI, the principal investigators at Stanford. And I think the answer to, well, our various attorneys will look at this and that will be decided.
And that's great. And that's why we love having patient advocates on these panels, because you ask the questions that maybe they hadn't thought about, but they need to as they get ready for a clinical trial. Well, that's all we have time for. Pat, thank you so much for joining us, for sharing your experiences with us, and for being on a clinical advisory panel. I know that the contribution you make, you downplay it a little bit, but it's really essential to make these projects as successful as they possibly can be. So that's all from us for now. We'll go into the cardiac stem cell session, but we'll be meeting more patient advocates later. So I'll see you then.
It's a pleasure to speak at the 2020 CIRM grantee meeting, and I should say before I start that all of the work that I'll present to you today was funded by CIRM over the last many years and would not have advanced to the point that I'll show you in the absence of that funding. And both stories rely on a cellular reprogramming approach to address human forms of heart disease. And in both cases, we've leveraged cardiac developmental networks to either regenerate damaged hearts by reprogramming resident cardiac fibroblasts to cardiomyocyte-like cells, or to understand mechanisms of disease using human iPS cells, and then followed by drug discovery. In the first story, we have leveraged the fact that the human heart is made up half of actually cardiac fibroblast and less than half of myocytes. And the cardiac fibroblast are the ones that support cardiac muscle cells, but also are the ones that are activated to form scar tissue, as you see in this section of this heart here. And because the human heart and mammalian heart in general has very little, if any, capacity to regenerate, once cells are lost after damage, as you see here, there's no capacity to regenerate. And so we over the years attempted to reprogram these resident cardiac fibroblasts into new cardiomyocytes right where they are in an effort to regenerate damaged hearts. And to make a long story short, over the years, we were able to find that the combination of these three key developmental transcription factors, GATA4, TBX5, and MEF2C, were sufficient to reprogram cardiac fibroblast into cardiomyocyte-like cells that we called induced cardiomyocytes, or ICMs. And this was relatively inefficient in vitro on plastic, but could be done. But in vivo, in mice, we found that this was quite efficient, and these resulting cells were most similar to adult ventricular heart cells. They could electrically couple with one another, which was key for improving cardiac output. And in fact, when measured by MRI, these mice did, in fact, have a significantly improved cardiac function. This is an example of what those hearts look like after coronary ligation, followed by a gene therapy-mediated delivery of these three transcription factors following 3 months later, one can sacrifice the hearts. And if you look at the apex cross-section around this level, you see abundant scar in the control, less so at the further up in the heart. And in comparison, mice treated with this gene therapy approach had abundant muscle even at the apex, as you can see here. And we have fluorescently labeled with a Cree-based system the fibroblast, and can see that all of these are actually newly formed cardiomyocytes. We, of course, asked, will the same combination work in human cardiac fibroblast? And it turns out that in human cardiac fibroblast, replacing GATA4 with myocardin, a transcriptional coactivator for MEF2C, actually was sufficient now to reprogram human cardiac fibroblast into cardiomyocyte-like cells. And you see an example of a beautifully reprogrammed cell here with these sarcomeres indicated with cardiac alpha actinin. And so this combination we then tested in vivo in pigs to see if which has a heart more similar to the size of humans, and in vitro this combination also reprogrammed pig cardiac fibroblast. And using an AAV now, a vector to deliver these three factors, we injected these into pig hearts after coronary occlusion and co-injected them with a retrovirus expressing DS-RED, because a retrovirus will only infect dividing cells, and myocytes don't divide, so it would allow us to mark the non-myocytes that were infected by virus, sometimes co-infected with the AAV, and then we can ask whether there are DS-RED positive cells that now have sarcomeres, suggesting that these might be newly born cardiomyocytes. And you can see here in this high magnification section that there are a number of cells with beautiful sarcomeres that are also red, suggesting that these may be newly reprogrammed cardiomyocytes. And you can see that a little bit more easily here with just the DS-RED channel in white. And so you can see that this is a fairly efficient reprogramming event in vivo. So this was very encouraging that we might be able to generate enough cardiomyocytes to actually make a difference. And this is in the border zone of the damaged area. And so with this information, all supported by CIRM, particularly the pig translational studies, we have put this technology into a startup company called Tenaya Therapeutics that was launched with the 50 million Series A financing with the Column Group in 2016, followed by a Series B event last year, and they are advancing this towards clinical trials. And over the years, they have refined the technology and the genetic material that would be delivered, as well as a vector. And I'll just show you one slide from Tenaya with their approval that shows you a cohort of pigs, around 10 pigs, either treated with the gene therapy AAV or control. And in blue, you can see that there's a marked improvement in the ejection fraction in these pigs compared to the controls in gray. And this degree of improvement of 11% absolute ejection fraction improvement is really quite significant and would be clinically meaningful, particularly for those who might be waiting on a transplant list to potentially be able to avoid a need for a transplanted heart. And so we're very excited about Tenaya pushing this forward, and we continue to collaborate with them to advance this technology. Now, in the second story I want to share with you, as I mentioned, it's really one about understanding disease mechanism and doing drug discovery with iPS cells. And this story relates to a family shown here where this multi-generation family had a very common form of heart disease called calcification of the aortic valve that looks something like this, where the aortic valve becomes hard and calcified, requiring replacement surgically. And about 100,000 replacements are done a year just in the United States, so it's a very common disease. The etiology hasn't been known, and there's currently no medical therapy. We do know that in cases where people are born with a congenital anomaly where there are only 2 leaflets instead of 3 in the aortic valve, like you see here, something we call a bicuspid aortic valve, that about a third of those individuals will develop calcification as they age into their third, fourth, fifth, and sixth decades of life. And this congenital anomaly is actually the most congenital anomaly of all. It affects 1% to 2% of the population. It turns out this family had both bicuspid aortic valve and calcification as they got older, and it's caused by a heterozygous loss of function mutation in the very well-studied transcription factor Notch1. And so, having identified the genetic cause of this, we were able to use isogenic CRISPR gene-edited, gene-corrected iPS cells from this family to deeply understand the mechanism. And it turns out that what we found is that normally Notch1, which sits on the cell membrane, is in a position to sense shear stress, and its job is to normally repress osteogenic pathways in the endothelial lining of the valve. And we know that the endothelial cells of the valve can transdifferentiate and become mesenchymal cells and go to the valve. And that's where this notch is playing a role in preventing this osteogenic fate. So essentially, what we found is that in the setting of haploinsufficiency, what happens is endothelial cells undergo more EMT and become more osteogenic. So it's essentially a cellular reprogramming event from an endothelial cell to a more osteogenic-like cell. And this we were able to discover through a deep interrogation of these iPS-derived endothelial cells, as shown here, that resulted in a deep understanding of the gene network that gets dysregulated. And it turns out that the network narrows down to 3 key transcription factors, SOX7, TCF4, and SMAD1, that are central players that then dysregulate a host of other genes that results in this cell fate switch, if you will. And so that gave us the thought that maybe we could drug this process. And so we screened a library of 1,600 highly curated compounds. And instead of looking for 1, 2, or 3 outputs, we screened for 120 genes in the network that were dysregulated with each molecule. And we used a machine learning approach to classify cells as either normal or abnormal and asked what drugs might reclassify abnormal cells. And from this, we found 6 hits that actually had resulted in this change in classification. I'm just showing you here what this output looks like. In blue, these blue dots are wild-type cells, normal cells. Green dots are what the machine learning algorithm classified as heterozygote cells. And in red are what the algorithm classified as either normal or abnormal. Here you can see that most of the drugs do nothing. They still cluster with the green and so are still heterozygotes. However, you'll see that there are a few red dots that are now clustering with the blue dots, and these are what our hits are, suggestive of a drug that is shifting this profile broadly. And so we have taken these 6 hits and tested them in vivo in a mouse model we generated, where by shortening the telomeres in Notch1 heterozygous mice to be more like human telomere length, we can actually recapitulate the human phenotype of a calcified and obstructed aortic valve. And so I'm showing you here by echocardiography in these mice, the aortic valve peak velocity, which reflects the aortic valve stenosis, very similar as occurs to in humans. This is partially penetrant, but you see in the control, a number of these mice have acceleration of the blood flow across the valve indicative of stenosis, both of the aortic valve and pulmonary valve. And one of the 6 drugs shown here had a remarkable effect in completely preventing in a statistically significant way aortic valve stenosis in the vast majority of mice, as you can see here, as well as preventing pulmonary valve stenosis. And by histology, this drug also prevented thickening of the aortic valve and calcification of the aortic valve. So we think we've got a drug that we discovered in human iPS cells that works in iPS cells to alter the dysregulation and works in vivo. Now, finally, we asked, would this drug also affect those who don't have Notch1 mutations as a cause? And to do this, we worked with the group in Russia who had collected primary aortic valve endothelial cells from explanted human aortic valve samples, either with 3-leaflet calcification or 2-leaflet calcification. And we exposed all of these endothelial cells to the drug and then did RNA sequencing. And I'm just showing you here the most important result of the most important factors, these 3 sort of master transcription factors that we know are causing the broad gene dysregulation. And in each case, this drug restored corrected the upregulation seen in the disease valves, endothelial cells, to normal, whether we're looking at SOX7, TCF4, or SMAD1. And it didn't matter whether they're tricuspid or bicuspid aortic valves. We saw the same things in the abnormal cells, and those were corrected by the drug. And so we're very excited that through this approach, we actually have a potential drug candidate, which would be the first that works in mice and works in broader populations of human aortic valve cells. And we're considering now how to advance this towards a clinical trial. So with that, I'll close and thank the members of my laboratory that contributed to this work, including former lab members and our collaborators, and of course, our important funding from CIRM. And with that, I'll be happy to take questions at the end of this session. Thank you very much.
Hello, my name is Mark Mercola. I'm a professor of cardiovascular medicine at Stanford University. It's a pleasure to talk to you about using induced pluripotent stem cells, or iPSCs, for predicting and removing drug cardiotoxicity. This work has been supported for many years by CIRM. We're very grateful for that work in establishing this platform in my laboratory. So what is drug-induced cardiotoxicity? This is the unintended adverse effect of medicines on the electrical and/or the mechanical function of the heart. This can be a very major problem for certain areas of medicine, such as oncology, where as many as 1/3 of the patients who have been treated with oncology drugs will develop some form of heart disease as a consequence of their treatment. It's also a major problem for pharmaceutical companies. It's the major reason for drug attrition, drug failure during the development, and in some cases, even after market launch. So we're using iPS cells to try to address this problem. So what iPS cells are cells that we can derive from your skin or your blood, any other cell type in your body basically, by reprogramming them back to an embryological state. This was first done by Shinya Yamanaka, based on work done by John Gurdon years before. And the technology for this won them the Nobel Prize for Medicine in 2012. So once we have iPS cells, which resemble cells of the early embryo, we can direct their differentiation to different cell types in the body, such as heart muscle cells, brain cells, kidney cells, intestinal cells, and so on. And so you are able then to study disease, because of course these cells retain the genetics of the original person, so if the person has a genetic disease, you hope that some manifestation of that disease will be reproduced in the laboratory. And we can also study the effect of drugs on those cells. So using this platform then, the work that I'm going to tell you about creates heart muscle cells, and you can see that in the video on the slide. We treat those heart muscle cells with drugs or drug candidates, and in order to predict the adverse effects that those molecules will have on the heart, as well as to understand the influence of patient genetics. Some people are much more susceptible than others to the adverse effect of drugs. And we also would like to use this platform to engineer safer drugs. So my talk today will be divided into 3 parts. I'll first talk about our recent work to develop efficient means of producing and increasing the fidelity of disease modeling in iPS cells. Secondly, I'll talk about using this platform to optimize an existing drug for a cardiac electrophysiologic disorder. And thirdly, I'll talk about our work to re-engineer an oncology drug to diminish its adverse effects. I'll start with the first part, producing cardiomyocytes efficiently and making better disease models. So when we began this work with CIRM funding back in 2008, it was only possible to make small numbers of cardiomyocytes, certainly nothing that could be used in high-throughput drug screens. So with grants from CIRM to John Cashman, a medicinal chemist at the Human Biomolecular Institute, and to me, and I was in those days at the Stanford Burnham Prebys Institute and at UCSD in San Diego, we developed a screening platform where we could look for compounds that would drive stem cells, in those days embryonic stem cells, to form cardiac muscle cells. And we hit upon a number of compounds that would do this. One of them is shown here. And these molecules are now the basis of nearly all efficient protocols to produce cardiomyocytes. More recently, we've been using this platform to advance the maturity of cardiomyocytes, iPS cardiomyocytes. These cells, if we form them by conventional means, are similar to cells of a very, very early embryo, for a few weeks, a month or so into gestation. And that hampers disease modeling, which of course we're interested in the adult. So we learned that if we switch the energy substrates that the cells burn, so rather than burning sugar, we switch them to fat and other substrates, now we can drive the metabolic, the structural, and the electrophysiological aspects of their maturation, and that improves their ability to model diseases as exemplified by dilated cardiomyopathy or an electrophysiological disorder, long QT syndrome type 3. So now, in the rest of the talk, I'm going to talk about our efforts to use this platform for drug reengineering. And the idea here is to make a better version of a drug that has some adverse effect on the heart of a patient that limits either the dosing or compromises the patient's health. And so the idea is that we would create heart cells from these people who have problems with some drug. We would then, in the dish, visualize the effect of that drug on the heart cells, and then we would, using high-throughput screens, using robotic platforms such as what you're seeing in the video, we would then develop better versions of those drugs, understand what to tweak in the drug to make it safer, and yet retain its activity. And that would then the idea would be to return that drug through a pharmaceutical development pipeline back to the patient. But the problem when we started this, it was a rather audacious goal, because iPS cardiomyocytes had never been used to drive a drug development campaign, certainly not patient cells where we were looking at a patient phenotype. So the question that we were all wondering is, will these assays in this platform be statistically robust enough to drive a medicinal chemistry exploration of a drug? And so we needed a test case. So with a Serum Early Translation 4 grant that was awarded to John Cashman and myself, we set about to re-engineer a drug that's used for a cardiac electrophysiology disorder, long QT3, and the drug was mixilotine. We also were grateful for the support of physiologists in New York and in Chicago, the labs of Rocky Cass and Al George. So what is long QT type 3? It's a rare genetic disease that causes ventricular tachycardia and sudden cardiac death, most commonly in teenagers and young adults. Its hallmark is that it has a prolongation of the QT interval on the surface electrocardiogram. Now, mixilotine will shorten that QT interval and thereby reduce the risk that the patient will develop a lethal arrhythmia. But the problem is that mixilotine at slightly higher doses than what's achieved in patients can also induce electrical problems in heart muscle cells. And although this isn't a huge problem clinically because these patients are very well managed, it nonetheless has induced concerns that it might induce or aggravate an arrhythmia. So even though it's not such a huge clinical problem, it's a wonderful test case for this platform because we can see the electrophysiological problems of cardiomyocytes quite clearly in these iPS cardiomyocyte assays. So we needed a patient. So Rocky Cass in New York and his clinical collaborators had identified a boy born with a particularly serious form of long QT3. He was actually diagnosed in utero and implanted with a dual chamber ICD that you can see here to control his arrhythmia. You can see the prolongation of the QT interval on the electrocardiogram, and he was treated with mixilotine and responded quite well. So we then made iPS cardiomyocytes from this boy, and we wanted to record their electrical activity. And it's much like what is done in clinic with an electrocardiogram, but we do it optically, and you can see the beating of the cardiomyocytes, and here you can see a normal electrical activity of cardiomyocytes, and here you can see arrhythmic activity where you get a prolongation of the so-called action potential, and you get these extra spikes known as early after depolarizations. So using this platform and using iPS cardiomyocytes made from the boy, we were able to run through libraries of structural analogs of mixilotine and identify what would be the good and the bad determinants in those structures. So we were looking for molecules that would shorten the action potential, but mixilotine on its own, when you give it at too high doses, will cause prolongation and these after depolarizations, and that's bad. What we were able to do eventually was find 4 molecules that would only shorten, but even at high concentrations would not prolong. So we produced a safer version of the drug. And this molecule has worked in studies to block the arrhythmia caused by long QT3. Now, emboldened by that, we now set out to do, which is really what I've always wanted to do, which is to re-engineer oncology drugs. These are, as I said, a serious problem. About a third of cancer survivors will suffer cardiac problems as a consequence of their treatment. It's not just old cancer drugs, but even the new molecularly targeted therapeutics that have this problem. We assembled a large team, co-directed by a medicinal chemist, Sanjay Malhotra, at Stanford, and Ioannis Karakakis, a stem cell biologist at Stanford. And these are the postdoctoral fellows, the trainees, who worked on this project. So the drug we focused on was ponatinib. It's used to treat chronic myelogenous leukemia because it inhibits the oncogene known as BCR-ABL. Now, BCR-ABL is in its normal form is inhibited by a relative of ponatinib known as imatinib. That is a safe molecule. But unfortunately, a large number of patients have a mutation that arises in the BCR-ABL gene, and that mutation renders the protein insensitive to imatinib and all other first-line defense drugs for CML. Ponatinib is the only effective drug against this mutation that's been approved. So whereas imatinib is safe, as I told you, ponatinib is cardiotoxic. 8% of people who have taken it have developed heart failure. Many more have developed heart disease. But since imatinib is safe, we thought the problem with ponatinib is an off-target effect, so we wanted to remove it. And the IPS platform is ideal for this because we don't need to know the actual reason why the drug is bad. We just need to know that it has bad effects that we can see in the dish. It's difficult to know the exact reason because these molecules target many, many proteins in the cell, at least 50 structurally related molecules to the BCR-ABL gene, and as many as 3 or 4 times as many non-kinase targets. So, what we did was we took the structure of ponatinib, we started tweaking parts of the compound in order to map the parts of the molecule that are important for its anti-tumor effects and important for the cardiotoxic effects of the drug. And we were guided by the structures of the relatively safe compound, imatinib. So we synthesized many analogs in an iterative process and tested them in parallel in iPS cardiomyocytes, in vascular cells, which form tubes, and in the tumor cells, both normal BCR-ABL and mutated BCR-ABL. And the idea was to define the structural determinants for the good and the bad aspects of ponatinib. And to cut to the chase, I'm going to show you 2 examples of improved molecules. And in this heat map representation here, red is bad and white is safe. And these are different indices of cardiotoxicity in the dish. Imatinib is white, so safe. Ponatinib is red, so bad. And these 2 analogs here, you can see are mostly white. And yet, when we can see that on the endothelial tumor vasculogenesis assay, you can see that they do not disrupt the vasculature, whereas ponatinib does, even at high doses. And yet they continue to block the growth of the tumor cells. The smaller number indicates better inhibition of the tumor. And so our new drugs, like ponatinib, will inhibit. And even the mutant tumors with a mutant kinase, they will also inhibit. So, and this works not only in vitro, those prior data were in vitro, but it also works in vivo if we have tumors, human tumors in mice. And by treating with ponatinib or with our new drugs, we can reduce the tumor burden substantially. So what I've told you then is that in parallel, using cardiomyocytes, vascular assays, and tumor assays, we could engineer a safer version of the CML drug ponatinib. We learned that there are different determinants for the good and bad aspects of the molecule, and that the new molecules that we've produced retain the anti-cancer effect, but have decreased cardiotoxic effects in vitro, and they have acceptable properties to go into animals, and they show anti-tumor activity in xenograft models comparable to ponatinib, but without the cardiotoxicity. This work, as I said, has been supported by Serum funding specifically for these projects throughout the 10-year history, as well as it's been aided by Serum training grants to the Stanford Burnham, the Scripps Institute, the Salk Institute, and the University of California at San Diego. I directed the training grant at the Stanford Burnham. It's also been aided by Serum infrastructure grants to those 3 institutions in San Diego. Thank you very much.
I am Deborah Lieu. I'm from UC Davis, from the Department of Cardiovascular Medicine. And today I'm going to talk to you about my research on the quest for pacemaking cardiomyocytes for engineering biopacemakers. So there has been a great interest in regenerative medicine for the heart because, as you know, cardiomyocytes or the heart muscle cells, and their stem cells, precursor cells, have very limited proliferative potential. And this is well documented by the study that shows the turnover rate is actually quite low, less than 2% after you're 10 years of age. So to replace these cells that are lost due to disease or age, there needs to be a source of human cells. So one possible source is human pluripotent stem cells. And these cells have the ability to self-renewal, and they can also expand indefinitely in culture and differentiate into any cell type in the human body, including cardiomyocytes or heart muscle cells, the infamous one being the human embryonic stem cells. But my lab only works with human-induced pluripotent stem cells that have the same self-renewal potential and the ability to differentiate into any cell type. But then these cells originate from cells from people's skin or blood, so they don't have ethical problems. And these cells are reprogrammed or reset back to this primitive state by overexpressing these combination of transcription factors that can reset the cells back to a pluripotent state. So once we get the iPSCs, or the human-induced pluripotent cells, how do we get to contracting cardiomyocytes? Well, we copy how the organs develop in embryogenesis by looking at the chemical environment during the development. So two well-established differentiation protocols from Dr. Sean Polachek's lab and Dr. Gordon Keller's lab. These protocols pretty much shows that if you take the iPSCs and you add BMP or activin A, or some kind of Wnt activating in the first stage, you can get the cells become mesoderm, and then followed by some kind of Wnt inhibitor in the later cardiac mesoderm stage, you can get these cells eventually become cardiomyocytes. However, the yield for these cells is not 100% cardiomyocytes. You get other cell types such as endothelial cells, fibroblasts, and on top of that, not all cardiomyocytes are the same. So you can largely categorize functionally. You can categorize the cardiomyocytes largely by their functions into pacemaking and contractile subtypes. So the pacemaking cells, they're really there to generate action potentials or to set the heart rhythm. So for applications such as to engineer Bio-pacemaker, you would need these cells to be able to self-generate action potential, and they tend to be smaller and have fewer myofilaments. On the other hand, if you want to replace cells for heart attack, you would need more contractile cells, and these cells tend not to have any ability to self-generate action potentials without outside electrical stimulus, and they tend to be larger and have more abundance of myofilaments to generate contraction to pump the blood. So based on our work, we have shown that you can get ventricular-like, atrial-like, and pacemaker-like cells from iPSC-derived cardiomyocytes. And we can identify these largely by their action potentials or electrical signature from these voltage changes from these cells. And then based on the phenotype, we can see that pacemaking cells shown by the orange bar is actually the smallest fraction of these subtypes. So how can we increase this pacemaking fraction? So something that we have tried is biochemical differentiation, looking at the biochemical environment. So one study has shown that if you eliminate this PX2 transcription factor, you actually can get development of another pacemaking tissue in the left atrium as well, as shown by the green arrows here. So we think that by knocking down this PX2 expression, transcription factor expression, then we can actually promote development of pacemaking cells in our differentiation. So we decided to block something upstream, the nodal pathway that controls the PX2 expression, by a small molecule SB431542. And so we added this SB small molecules in a stage-by-stage manner to test along the differentiation timeline. And then we found that when we add in the cardiac mesoderm stage from differentiation day 3 to 5, we don't see an adverse effect on the differentiation as assessed by troponin T expression, which is an indirect assessment of differentiation efficiency of cardiomyocytes. And then we also can see from the video below, comparing to the control cells shown on the left, when we added SB, you can see the contraction rate is actually much faster, and the contraction amplitude is actually smaller. And this is good sign that we're getting more pacemaking cardiomyocytes. And then we did some flow cytometry to assess the cells. So first we looked at the yield of cardiomyocytes, which can be determined by the number of troponin T positive cells, which is shown on the box on the top side of all the plots. And then you can see that when we added SB in both IPS lines that we tested, there's actually an increase in the yield of cardiomyocytes as shown by the red bars. And these cells are actually smaller than the control cells, and that's an indication of pacemaking cells. And we also looked at some pro-pacemaking transcription factor, T-box 18 and T-box 3, and you can see when we added SB, it shifts the histograms to the right, and that indicates that there's a higher expression of TBX18 and TBX3. And we quantify these increased expression in 2 different cell lines, and you can see relative to normalized baseline, there's increase in the expression of these transcription factors after we added the SB molecule. And then next, we verified the protein expression of SHOX2, pro-pacemaking transcription factor SHOX2 showing green, and also IL-1 showing red. And when we added SB, you can see there's increase in staining in the nuclei of the iPSC-derived cardiomyocytes. And similarly for T-box 3 and 18, there's also increased expression in the SB differentiated cells. And then the ultimate test is really functional test in these cells. So we looked at the action potentials of these SB differentiated cells relative to control cells using whole cell patch clamp that measures the voltage of the cells. And we also did this with another method that recorded the cell optically with the voltage sensitive dye. So I won't go too much detail into the data, but you can see the action potentials from the SB differentiated cells are actually have more action potential that resemble more like pacemaking cells compared to the control cells. And another factor that could possibly affect cell characteristics is really the microenvironment, or the extracellular matrix that surround the cells. So extracellular matrix is basically a network of extracellular matrix protein that can create this scaffold that surrounds the cell. So we hypothesize that the extracellular matrix, or ECM, from the pacemaking tissue or the sinoatrial may be a more suitable microenvironment to promote and maintain the pacemaking phenotype in the iPS-derived cardiomyocytes. So we took a stepwise reverse engineering strategy to first look at what is different about the ECM in the sinoatrial or the pacemaking tissue. So we harvested the tissue from a pig heart, and we identified the pacemaking region by a region that's negative for this contractile marker connexin 43 and positive for this pacemaking ion channel HCM4. And then we decellularized, which means we chemically extracted the resident cells out to leave only a matrix scaffold behind. And then we assessed the ultrastructure of the matrices, and we found that the sinoatrial matrix have these more rope-like fibrils compared to the left ventricular matrix. And when we measured the stiffness of the sinoatrial, and that's quantified by this stiffness factor, Young's modulus, you can see the sinoatrial matrix shown by this distribution of moduli, it's actually higher or more stiffer than the left ventricular matrix. And we also did mass spectrometry to look at the biochemical composition of the proteins in the matrix. And in general, there's more abundance of fibrillar collagen in the sinoatrial node compared to the left ventricle. And also there's actually abundance of elastin as well, stretchable elastin as well. And we also did immunostaining to look at the distribution of some of the major extracellular matrix proteins. So based on our findings, we came up with this hypothesis that if you consider the sinoatrial matrix as a composite material, then the pacemaking cells are actually enclosed in this stiff tensile bearing fibrillar collagen region, while there are elastin fibers that spans the stiff region. So when the heart is undergo active contraction, the elastic region will actually undergo deformation more readily than the stiff region, and then this will protect the cells inside the stiff region from mechanical strain compared to the cells that are actually in the left ventricular matrix. The cells will actually experience more strain because they're not as well protected by the matrix. So this is just showing a force plot. We think the force is actually higher imposed on the cells in the contractile region than the pacemaking region. So we decided to test to see if we can use the sinoatrial matrix to better maintain and protect the pacemaking cells. So we took the porcine heart, we again decellularized the sinoatrial matrix and also the left ventricular matrix as control. And then we differentiated our iPSCs to cardiomyocytes. And then we recellularized or basically seeded the matrices with our iPSC-derived cardiomyocytes. So now we have recellularized sinoatrial heart tissue, and also for control, we had recellularized left ventricular heart tissue. And in the end, we'll actually transplant this into the diaphragm of immune-compromised mice to basically use the mouse as a bioreactor to subject these tissue constructs to cyclic mechanical contractions at a rate that's similar to a fetal heart rate, but without any electrical impulse interference from the resident muscle cells, because the skeletal muscles from the diaphragm do not electrically couple with the cardiac muscle cells. So we don't have to worry about any electrical interference from the skeletal muscles. So right after recellularization, we noticed that the iPSC-derived cardiomyocytes, showing the red, that were seeded onto the sinoatrial matrix, they organize themselves into clusters, very much like the native sinoatrial at the very top. Versus those cells that were seeded onto the left ventricular matrix, you can see they form these aligned layers, very much like the native left ventricular matrix. And then we also did some functional measurements looking at the calcium dynamics using this GCAMP calcium sensitive probe that look at the frequency of calcium changes. And then what we found is the rate of changes in these the cells that were seeded onto sinoatrial is definitely much faster than those on the left ventricular matrix, as shown on the bottom right graph. And the amplitude is actually also smaller. So these are traits that are indicating pacemaking-like cardiomyocytes. And then we transplanted the sinoatrial and the left ventricular recellularized tissue constructs into the diaphragm of the mice. And then we see even after 2 weeks of cyclic mechanical strain in the mouse, we can still see the iPSC-derived cardiomyocytes shown by the red still clustered or organized like that in the native sinoatrial, and those in the left ventricular matrix, those cells actually stay aligned after 2 weeks. And then we looked at the pacemaking markers in these iPSC-derived cells that were recellularized onto the sinoatrial matrix versus the left ventricular matrix. So for pacemaking markers TBX3 and TBX18, there's definitely higher expression of those transcription factor in cells that were cellularized into sinoatrial and versus the left ventricular matrix. And the pacemaking channel HCM4 is also has higher expression in the iPSC that were grown in the sinoatrial matrix relative to the left ventricular matrix. And then the contractile marker connexin 43 is actually higher in the left ventricular matrix. So all these are indicating that we have a more of a pacemaking phenotype in iPSC cardiomyocytes that were cellularized in the sinoatrial node. And the last thing we did was, again, to look at the functional aspect of these cells. So again, we used the GCAMP calcium sensitive probe to look at the calcium dynamics in these cells. And as you can see in this graph, that shows the frequency of automaticity in the iPSC-derived cardiomyocytes in the sinoatrial matrix is much faster than that in the left ventricular matrix. And also the amplitude, again, of the calcium transients is actually smaller. So again, this indicates that the iPSC cardiomyocytes seated in the sinoatrial matrix maintained the pacemaking phenotype better. So the take-home message is that pacemaking cardiomyocytes can be derived from iPSCs, and we can direct the subtype by looking at the chemical environment, such as playing around with the nodal signaling that I talked about, and also providing the right microenvironment, such as the extracellular matrix from the pacemaking tissue. And then stem cells really opens the door to possibility of engineering bio-pacemakers from human pacemaking cardiomyocytes. And I would like to acknowledge all the people, past and present, from my lab, and also my collaborators, Drs. Nipavan Chami-Monvat, Dr. Xiaodong Zhang, and James Chen. And this work has been funded by a CERM Discovery Grant to the Lieu Lab. Thank you.
I guess perhaps while we're waiting to get some questions, I might start off by asking Deepak, if one came up with some drug that could in fact alter the potential calcification of aortic valves, would you anticipate giving this early in life to everyone who had a bicuspid aortic valve?
Yeah, I think that's a good question. As I mentioned, about a third of patients with the bicuspid valve will get calcification over the coming decades. So it wouldn't be, if it's a relatively safe drug, it would not be unreasonable to do that as a prevention. I didn't mention, but we also did test in mice this drug in situations where the calcification and aortic valve stenosis was already present, and asked, does the drug either slow down the progression, which would be good in of itself, or actually completely arrest it? And in a small number of mice we've treated, the drug actually arrests and in some cases even reverses the aortic valve stenosis. So I think in high-risk groups, I think prevention would be reasonable. But what's even more encouraging is that I think even if calcification is detected, usually we just then observe it over years, and when it's bad enough, we operate. And in this case, if you just stopped it, most people, I think, ultimately would not need any surgical intervention.
Yeah, no question. My patients who have a modest or mild degree of aortic stenosis are always asking me what they can do to prevent the progression. And to have a drug that would do that would be such a colossal advance. I can hardly wait to see how you move forward with that one.
Thank you. Thanks to CERM. Wouldn't have happened otherwise.
Great. So perhaps Dr. Lieu, while we're waiting for some additional questions, I might ask you, are you envisioning a bio-pacemaker as a patch? Some kind of a fully organized tissue that would be implanted into the heart?
Yeah, that is what we're envisioning, because I think if you inject the cells into the myocardium, it will be subjected to this mechanical strain. And then we know the microenvironment can actually affect how the cell phenotype and also the cell function. So we think we need to implant basically the pacemaking cells in some kind of scaffold that can protect the cells from this mechanical strain once you transplant it into the myocardium.
So just a kind of interest, hopefully we think of some kind of catheter procedure that could do that, because most patients might say, I just as soon have the pacemaker.
Right, yeah.
I'm sure you can do that along the way. So Mark, nice work, even though you escaped from San Diego. Very good work. So virtually all these wonderful cancer drugs that are being developed do have an incredible amount of cardiotoxicity. It's a huge factor. There's a high percentage of, for instance, women with breast cancer who die of heart failure from the drugs that they're given to treat them. How do you see proceeding with the nice work you've done with the drugs that you studied?
So I think the easiest class of drugs to deal with are those for which the reason for the cardiotoxicity is due to an off-target effect. And as I said, it's hard to know the off-target effect of any of these drugs causing the problem. So that's why we use this phenotypic assay to drive the derivation of newer versions of the drugs that are safer. So a drug like ponatinib, for instance, that we believe it to be an off-target effect, I think this could be taken forward through a pharmaceutical company development pipeline back to patients, and that's what we're attempting to do. I think many drugs could be tested early in their development. Cancer drugs tend to be tested rather late in their development for cardiac effects, and I think that, and as a class, that perhaps is one of their problems. So testing early so that this platform could be implemented to avoid these problems in the future. that would be helpful. But I think in terms of just taking a new version of an existing drug forward, I think that's the one of the simpler ways to move back to patients. For the drugs for which is an on-target effect, that's a lot harder to deal with, and just tweaking the drug it's less obvious how to do that, and so it's easier for the drugs like the kinase inhibitor drugs, for which we believe in many cases it can be you can change the target space and improve the quality, improve the safety of the drugs.
And your screening technique is really impressive. Is that hypothesis-based at all, or do you have just a huge number of potions that you put?
No, it's very much hypothesis-based. So, we screen iteratively about 20 new compounds a cycle, and we go through, say, for the Mexilatine project, the test case, we went through about 180 compounds, but going through about 15 to 20 with each cycle. So each cycle you make a guess about what you think you need to tweak, and then as you go through these cycles, you learn whether one part of the molecule provides a determinant that causes, say, a desirable effect or the undesirable effect. And so you slowly try to prune the molecule. So it's not as if we just randomly screen, a gazillion drugs to look for a better version. No, we're taking in this re-engineering approach, we're taking an existing drug, looking at that molecule, looking at what's known about it, and then starting to probe different parts a little bit at random, and then ask, have we tweaked the good or the bad effects, trying to preserve the good, and then eliminate the bad. So you can find, hopefully, different regions of the molecule, different chemical determinants in different positions that are responsible for the good and the bad effects.
Yeah. So Deepak, I might go back to you. You've got a drug that's entering clinical trials, and there's always this tension as to when we are dealing with some kind of a stem cell preparation, when we're ready to go into clinical studies. How do you make that decision?
Yeah, we talked about two different programs, and one that is advancing towards clinical trials is actually a gene therapy approach that is being advanced with Tenaya. And so that, I think, will await some further large animal studies and tox studies. But you might have been referring to the drug that I mentioned. That we're exploring right now. I think, we've discovered it in human cells. We've tested it in vivo in an animal model, rodent model. There aren't a lot of other models in which to test this before it goes to clinical trials. So we're right now trying to think deeply about what the best path would be. Obviously, there's some chemistry that has to occur. We used a tool compound that would not be the one that would go into a clinical trial. So there's probably mostly chemistry refinement that needs to be done. But then at that point, I think there's not a lot more experimentally that we may do other than testing in different conditions. For example, we are testing whether this vascular calcification, which is very similar pathogenesis as valvular, whether this might actually prevent also vascular calcification. Similarly, in infants, which I'm a pediatric cardiologist, infants are sometimes born with a very thick aortic valve when it's bicuspid, even without calcification. This drug also, in mice at least, affects the thickness. And so we're testing whether in neonatal mice that have a more severe form, where we have a model, if it can prevent that thickening in neonatal aortic valve stenosis as well.
Sure. Now, I assume that you don't have an MBA, and in terms of translating a discovery into a clinical product, there's a lot of steps along the way, and in terms of getting approval and getting reimbursement, even getting the funding to do the clinical trials. Have you assembled a team, or how have you gone about this? Has CIRM helped you?
So CIRM has helped us do all the preclinical work that I mentioned. And it would be really valuable if to be able to have CERM support, as they've done in other cases, with actually the team that you mentioned. I think that's one of the greatest contributions of CERM, in addition to the financial support, was actually the skill sets necessary to translate an idea into a drug through their disease teams and those sorts of efforts. So you're absolutely right. That's not our sweet spot. Our sweet spot's discovery. And the kinds of efforts CERM has been able to support in the past has been extremely valuable in that translation. So we're searching for that exactly right now.
Right. So Dr. Lieu, I'm impressed. I remember the early days when generating iPS cells was just gee whiz, and you know, it's seems to be translated to a so what now. I mean, I just generate a whole bunch of iPS cells and conduct my experiments. Is that the state of the art at the moment?
Definitely, the process has much simplified with improvements in how we can culture these cells. You know, because before, when I first started, we have to grow these cells on mouse fibroblast feeder layers. So that was a lot more work. Currently, we can just grow them on a matrigel, and the media that people have developed is also definitely a lot more supportive of the iPSC culture. But then I think the differentiation process for these cells has definitely moved. You know, so initially it was just to increase the efficiency of just generating cardiomyocytes from iPSCs, and now we're finding out not all the cardiomyocytes are the same, and you really need a specific type of cardiomyocytes to treat a specific problem. So we have refined this process of differentiating cells into a specific type of cardiomyocytes. And I also see in a chat box that somebody asked about the number of pacemaking-like cells that we get after addition of the SC molecule. So in general, we see about 2 to 3-fold increase in the number of pacemaking cardiomyocytes. And we are not sure if this number actually further increases after in vivo transplant in mice, because that is a little bit hard to quantify. But given that the nature of these immature cardiomyocytes, they have proliferability, we think there definitely could be proliferation of our iPSC-derived cardiomyocytes in vivo, because we think there may be vascularization of our construct going on that's actually helped the culture of these tissue construct we're generating compared to just the in vitro condition. But there could be some cell death as well associated with just the transplant, because initially they're transplanted into this environment without vasculature, so it becomes very nutrient depleted initially. So we're not sure where the balance is at this point. Yeah.
Thank you. So Deepak, we have a question here as to whether there are additional mouse models of aortic defects besides Notch.
There are a couple of other mouse models where the valves do get calcified. And so that is an interesting question, and we have not tested the drug that I mentioned on those animal models, but that would be an interesting thing to do. We actually went directly to the sporadic human patients who did not have a Notch mutation, and I think the key point is that we are searched for a drug that didn't affect Notch, but rather the whole gene network that was downstream that would occur in a calcified valve. And so we're not so surprised that even 20 different patient samples that it worked broadly in those. And so we haven't tested other mouse models, but we could.
So I might turn to Mark. You mentioned something about trainees and training grants. I'm impressed to see that all of you have a large number of trainees. Are these by and large becoming independent investigators, and is CERM responsible for helping in that regard?
I think that's one of the enduring aspects of the CERM funding over the last decade, is that we've trained a lot of people under the auspices of training grants at all the different institutions. And I think, you know, these were some of the best of the trainees that we had in the stem cell area. They competitively applied to these programs. So I don't think it's a surprise that they've gone on and done fairly well. So yes, I think we can all cite that they have, good examples of them going on and seeding stem cell research throughout the state and beyond. I think, as Deepak said, I think a benefit of the serum grants was that they were able, they sort of fostered us pulling together these diverse teams, and the trainees also then learned how to interact with people of different disciplines. In my area, of course, there were engineers dealing with physiologists, dealing with stem cell biologists and clinicians. So I think that was an important part of their training.
Okay, we have a question here for Deepak. Does in vivo reprogramming cardiomyocytes have a synchronization issue due to heterogeneity?
Yeah, that's also a good question. What I can say is that we have looked to see whether the reprogrammed cells can electrically couple with one another, and experimentally we can do that and see that they do. We've also looked, still been concerned about the heterogeneity that might occur in cellular states that would make some more excitable than others, and so have done telemetry both in mice and in pigs, and looked at a lot of EKGs, and do not see evidence of arrhythmias with the cocktails that I described in any of those models.
Great. Another question, I'm not sure for who, but many kinase inhibitors induce death of cardiomyocytes besides EPHIS changes. Has there been a consideration of screening drug-induced cell death or damage of cardiomyocytes?
Yes, so in fact, for the cancer drugs, I didn't go into this in detail, but for the cancer drugs, we screen not just the electrical, but we're more focused for those drugs on the cardiomyopathy effects. So one of the most sensitive readouts for many of the cancer drug effects is a loss of the contractile power of the cells. So that's some change in the contractility of the cells. We see that before we see cell at lower doses than cell death typically.
Can you do dose ranging studies?
These are all done in every screen I've talked about is done with typically 8-point dose curves and in replicates and in multiple differentiation batches of the cells, and also for the toxicity studies in cells from different people. So they're pretty comprehensive studies. And we see patient to patient, person to person differences. That was a question that's sent to me on the chat, so I'll answer it online, live. Even among healthy donor, iPS myocytes, if we expose them to a panel of cancer drugs, for instance, or other drugs, we can see differential effects among healthy donor. You can then, if you look at this on the same genetic, you could say, well, that could be due to lines in the genetic backgrounds. If we take the same genetic background, we introduce disease-causing mutations that can sensitize to certain classes, but not other classes of drugs. So you can see both genetic background effects as well as disease-causing gene variant effects.
Okay, well, we're in the closing minutes of this cardiac program and our discussion. So perhaps I'll ask all three of you for your last take-home thoughts about future directions in cardiac cellular therapy.
There's a tremendous need, number one, that's evident as heart disease remains the #1 cause of death. I think that's key to remember. I think there's a lot of potential for regenerative medicine, whether it's through cellular transplantation that's advancing still, or cellular reprogramming approaches. And then I think you've heard many instances of how drug discovery and drug toxicity things can be enabled by stem cells, particularly cardiac stem cells. And so I think a lot of that will result in a more tailored medicine, greater precision medicine, and our ability to maybe let cancer take over and be #1 and heart disease be #2.
I
Mark?
would just echo that. I think that's very well said. I think from using the cells as a therapy, or teaching you, like Deepak has done, about how to generate, how to develop or stimulate an endogenous regenerative response, I think that's certainly was at the core of the serum mission. I think the using the cells like we're using in drug discovery, I think that's that opens up a lot of new doors. It brings the patient early on into the pharma pipeline at the very beginning, right? So you have people and their diseases up front. So you're treating basically a realistic human model at the very beginning, not a reductionist model. So I think it's quite valuable to bring the stem cell technology. And then of course the straight modeling and putting in the tissue, like Deborah talked about, I think that that's what we had all thought would work for regeneration. Might not be so great for myocardial regeneration, as Deepak pointed out, but for specific tissue types and putting it in, like Deborah was saying, I think that sounds very exciting.
Dr. Lieu?
Yes, I definitely think the stem cell plays a huge role in regenerative medicine and also just drug development in general. And I definitely think there's a need, for STEM type funding, because this is more product driven and very different than how the NIH funding agency is focused on. So there's definitely a great need for product driven type funding.
Well, as Deepak suggested, cardiovascular disease continues to be the #1 killer in our society. And while we've come up with incredibly effective devices, there's a need for more pathophysiologically oriented approaches to disease management. And cell therapy, in any of its multiple forms, really, I think, is the most exciting direction to go in. So I want to thank the panelists and bring this session to a close. Thanks very much.